Hello, my name is Ian Thompson. I'm an instructor in medicine at Harvard Medical School and an associate biochemist at Brigham and Women's Hospital. I'd like to take this opportunity on behalf of myself, the co-authors, and the senior author, Professor Ursula Kaiser, in whose laboratory this study was conducted, to thank the Endocrine Society for allowing us to present our work, which was recently published in Molecular Endocrinology, entitled The GnRH Pulse Frequency Control of FSH beta gene expression is mediated via ERK1/2 regulation of ISA. Now, the overarching goal of this project is to elucidate the signaling pathways activated by GnRH in the anterior pituitary gonadotroph cell. So GnRH is released from the GnRH neuron in the hypothalamus in a pulsatile manner at either high or low pulse frequencies, and this in turn stimulates the synthesis and secretion of LH or FSH in the gonadotroph, respectively, dependent on the pulse frequency. In a publication released in 2013 in Molecular Endocrinology, we showed that at low GnRH pulse frequencies, the PKA pathway was activated and stimulated the phosphorylation of CREB to induce FSH beta transcription to a greater extent versus high GnRH pulse frequency. CREB, the cyclic AMP response element binding protein, is a transcription factor that binds to a half-Cre AP1 site in the FSH beta promoter that is conserved in humans. The main focus of this study, ISA, the inducible cyclic AMP early repressor, competes with CREB for binding at this half-Cre AP1 site and has been shown to be induced at high GnRH pulse frequency and acts to repress FSH beta transcription versus low GnRH pulse frequencies. So as the data show in our manuscript, under tonic GnRH conditions in the albidity to gonadotroph-derived cell line and dispersed mouse primary pituitary cultures, we see an induction of ISA by GnRH, and that this effect is inhibited by a MEK1/2 blockade. Moving to the more physiologically relevant perfusion paradigm, as expected, we see an induction of ISA mRNA and protein at high versus low GnRH pulse frequencies, and indeed in the presence of the MEK inhibitor at the high GnRH pulse frequency, the induction of ISA is abrogated. So now, having reduced ISA induction at the high GnRH pulse frequency, we would expect perhaps an increase in FSH beta transcription at this pulse frequency. And as our results show in figure 6, this wasn't actually borne out by our results. In fact, we saw an inhibition of FSH beta expression in the presence of the MEK inhibitor. And there are several potential reasons for this, and one that we addressed in figure 7 is that there are other AP1 proteins, such as CFOS and c-Jun, that are induced by GnRH that are promoters of FSH beta transcription. And these, in turn, were also inhibited by the MEK blockade. So not only did we reduce ISA induction by GnRH at high pulse frequency, we also reduced the induction of CFOS and c-Jun. And this could be a potential explanation as to why we didn't see the effect on FSH beta transcription as expected. So in terms of future work, we think it would be interesting to see what the effect is of either global or gonadotroph-specific ISA and CREB knockout mouse models. The effect would be on fertility, and their reproductive phenotype. So finally, I would like to thank again on behalf of the authors, the Endocrine Society, for allowing us to present our work. Thank you.